Thank the organizers of this conference, Joe and Sue, for the opportunity to present here, and also the help of an unknown individual who helped make this presentation possible. I, like the previous individual, always knew about the Shroud of Turin and thought, wow, wouldn't that be great to do some DNA analysis on the Shroud, because that's potential. And most of my work at the University of Utah actually involved ancient DNA analysis of the Anasazi and the Fremont Indians, and we've since worked on some sites in Egypt and that type of thing. So among those travels, the way I came to this is I was asking a collaborator and an individual to please validate some work that we're trying to do on the identification of the unknown child associated with the Titanic disaster. And he said, sure, but you're going to owe me a favor. He said, there may be something, we may get some samples from the Shroud of Turin, and maybe there's a chance here to validate some DNA. I thought, right, like that's going to happen. So I filled out the papers and sent them off and didn't hear anything until a package came in the mail from Julio with a sticky tape sample. And so I quickly did all of the due diligence that I could on the Shroud of Turin and read Ian Wilson's book and DNA of God and all those types of things. And one thing that really strikes me after listening to all the talks today and the controversy over the carbon-14 dating and all the work that's been done, and it was really made in the last presentation as well, is that sample acquisition is really key. If sample acquisition is not done correctly, then you get a bad result. And a bad result is really worse than no result at all. And we've just been dealing with the FDA on some issues that we have with our business. And there's a lot of scrutiny that goes into these things. So, so the science must be done with the proper controls and with replications and that type of thing, or it absolutely will not pass peer review or the scrutiny of the public. So that's when I first saw the sample that was sent to me, that was sort of the consideration that came up. I know one of these buttons works. Am I as bad as the last person? No.
I don't know. Are you a PhD? Forward.
Oh, forward.
Backward.
Oh, there was no place to go backwards.
That's right.
Maybe you should just stand here.
I'll be right there.
Okay. Okay, good. So, in my reading, it became obvious, and we just heard that there's a lot of iron associated with the Shroud. And one of the problems with DNA analysis is that iron is also associated with blood. So, of course, you have to be able to convince yourself that the iron actually is indeed associated with blood. So what we see on the Shroud, is that really blood? Or as others have claimed in Ian Wilson's book, is that just simply pigment from whatever source? I guess there are many sources that it could be from. So that was the first thing that we wanted to test. And then, so this is really a talk about DNA that got lost in analysis to really demonstrate, is there something here that's useful that we can move forward with? So then the second part is just really recommendations about DNA analysis and approaches and how that could be done. So what we did is, after having visited the FDA, it was very clear that there had to be comparables and that there have to be controls. And again, that was emphasized in the last talk. There have to be these comparables that convince you that what you have is really what you have. And so we picked up some controls. Of course, I had some colleagues who sent me many samples from the Fayoum in Egypt, and that's an area where there's a Coptic Christian site, and they had some burial linens. I didn't go so far as to really see whether or not they're from linen or cotton, but I believe that they were linen. And then we did I'm sorry, but this was irreverent, but we did what we called the Shroud of Brian. Brian was the individual that was doing some of the work that I'm going to talk about, and we wanted a modern comparison to blood. So he bled on some common kitchen textiles, and then he sweat on the linen, and we ran that through our analyses. And then just for a plain control, there was just a bleached linen. So This isn't linen per se, but it's just probably cotton. So that's how we went about this. Okay. So the origin and history of the Turin Shroud sample that was sent to us. I'm sorry, I'm not a shroud person. D. Numana aspirated the dust between the back of the Turin Shroud and the Holland cloth also in 1988. So as we saw yesterday from Giulio, the aspirated dust are conserved on filters. A sample was given to Giulio, and he selected a potential blood particle with a stereo microscope and a petrographic microscope, and then he put them on slides. And again, these were sticky tape samples, and the sample was likely removed from the foot area of the Turin Shroud. So this is what the sticky tape actually looked like, the sample. So I kind of refer to it as an inverted comma. And here's all of the material of interest, really this red area, because of its signature was believed to be a good possibility for blood. Well, the thing that we did, we decided to try SEM-EDXA on it, which will actually identify the components that are in this sample. So the next thing we did is we put it on double-sided or double sticky, so this is still a sticky tape sample, on tape in preparation for electron microscopy and EDAX, and here's what the sample looks like. So it clearly looks quite different from the previous piece, and you can see that everything now has a very crystalline look to it. So the object of our investigation then would be to investigate what these crystalline structures, what their chemical signature would be. The next sample, so I'm sort of going to go through the detail of the samples here just to show you that we tried to go into a lot of detail on this. It's really from the Fayoum, from a cemetery called Fag El Gamas Cemetery. And the Fayoum, if any of you are probably aware, is in Upper Egypt. And in this particular area, this is a Coptic Christian site. And so we're looking for materials that would be roughly contemporaneous, that would be comparables, dating to the same sort of approximate times. So this is the sample that we were granted. It was excavated in 1998, and the artifacts at this particular site date between 200 BC to 800 AD, and it's an interesting area because many of these fabrics don't look like normal, I guess you could say everyday Coptic Christian textiles, but it looks like they're a transition period between Pharaonic and Coptic Christian periods. However, this linen fragment likely dates between 200 to 300 AD. So the point of interest in this particular fragment, I asked them to send me something that was red, are these threads, these individual threads, are they pigmented with iron, which might have been a practice in this particular time. So we took out actually these little threads and mounted those on a stub and did the analysis. Controls, again, the common kitchen textile, saturated with blood, dried at 45 degrees centigrade, 8 hours, bleach, and perspiration. So here's a look at electron micrograph of what this looks like. So then we tried SEM-EDXA. And had I known that Los Alamos was doing some of this, would have saved us a lot of trouble because they do all this for free, apparently. So I'll have to talk to you afterwards. So my understanding of how this works is that an electron beam is aimed at the sample, and then the sample, don't want to go into the electron orbitals and all that type of thing, but an X-ray is given off and it's detected and it's counted, and then there's a chemical signature from that that looks like this spectra. So you get the same sort of data as we just saw. So peaks mean you have more, and here's the spectra along here. Here's what the apparatus looks like and has this nice electron beam facility there. So So again, I'm kind of jealous because I don't have all these really nice, cool-looking color spectra. So we'll have to talk about that as well, how you do that. So with some guidance from Giulio, we began to look at these particular areas. And what's interesting is that right away you'll see sodium here and chloride, sodium and chloride. And actually this zinc is a contaminant that they always get in this particular laboratory. But if you look over here, there begin to be spikes of iron. So indeed, olive and here as well. So indeed, the idea that the shroud has iron is once again proven. The results B, just more of the same, sodium, chloride, iron, telling us what we already know from the last result. And now here is the SEM-EDXA from our comparative sample from the Fayoum in Egypt. And you can see that it is really quite different. There's calcium and there is some iron, but not very much. And so we actually looked at several sites on here, but this piece is interesting because this is just looking at one of the threads or whatever's left once we tease it apart. And there is hardly anything. So are these guys contaminants? Are they dye? Are they pigment? If they are, and it's iron-based, there certainly does not seem to be a lot. So right now, it looks like a comparative from the same period is significantly different. And what's very important, you'll see in the next, is when we start to look at the modern blood samples, what you start to see this sodium signature again, and you see the iron signature. So these are two things that modern blood and the shroud now have in common. So the results, the Turin Shroud, it has the sodium chloride and the iron. What's interesting is it looks like it is a degraded sample because for sodium and chloride for table salt, the ratio between those two atoms is 1 to 1, but it looks like that's not quite the case as it is in modern blood where the sodium and the chloride are equal. However, there is this iron. The Fayoum linen has some iron, but it's quite a bit less than the Turin Shroud, and it also has some calcium. This is interesting, which is a bit of a side note. When I spoke to the collaborators at Brigham Young University that supplied this, we're wondering if this is sort of a shadow of calcium carbonate. So was calcium carbonate really used in the burial process anciently like natron was? And calcium carbonate may be the Fayoum's version of natron. So if you look at bleach and sweat, there's absolutely nothing. So I think what this tells us is that there's very good reason to believe that what I was seeing on the sticky tape is actually blood. So now, let's sort of move on to the Turin Shroud DNA analysis, just sort of an approach. So what would we want to do? What are the expectations if we were going to run some DNA analyses on the Turin Shroud? There's a couple of archives of genetic information in the human body. There is the nuclear DNA, and this is what you've heard a lot about recently. Groups like 23andMe have been in the news. And also, so nuclear DNA is a little difficult to use for genealogical purposes because you have a combination of your mom's DNA and your dad's DNA, and it's shuffled every generation. Mitochondrial DNA, is quite different because you inherit that in the ovum of your mother. So it's actually clonal from generation to generation. And in the simple genetic alphabets of G's, A's, T's, and C's is actually written a maternal name. So you can do maternal genetic genealogy just by using your mitochondrial DNA. And it's a very good tool for ancient DNA analysis because there are thousands of copies in a cell, as opposed to the nucleus, which just has one copy from mom, one copy from dad. So there are advantages to that. So this is typically what we'd use in forensic work or ancient DNA analyses. And in fact, in our work on identifying the unknown child, it was mitochondrial DNA, the unknown child from the RMS Titanic disaster. It was actually the mitochondrial DNA that was able to make the exclusions. So the maternal line, as I've already mentioned, you inherit it exclusively from your mom. If you're a man, this is where I'm sure that Mother Nature is really a woman, because men are discriminated against on a genetic basis. We don't pass on mitochondrial DNA. We inherit it from our mom, but it's not passed on. However, we get even with the Y chromosome. So this is what we'd probably want to look at in our expectation for what we might think a person from the 1st century AD might look like. We could look for certain patterns in the Y chromosome. So the Y chromosome is, of course, inherited by men. So we have a maternal signal that we could look for and a paternal signal. So what would the expectation be for someone from that particular time period and provenience? The literature tells us that there's actually indeed a mitochondrial maternal name that we've now called haplogroup X1. So mitochondrial names or Y chromosome names are always referred to as haplogroups because they're not specific, but they cluster together. So there is this X1 that was known to be in the ancient Near East, anciently. So as populations sort of subsist, and this was the main hub, and I don't think people were coming over from the New World or anything like this, I would expect to see this maternal haplogroup, this X1. And I also would expect to see the Cohen-associated Y chromosome. Now that doesn't mean that if you don't see these, that the person isn't of ancient Near Eastern ancestry, because remember that, well, 50% of Cohen Jews have this Y chromosome today. There are still 50% of them that don't. And the European populations look quite different, so I think that that would give us some hope in teasing out the differences between contamination, and I have to think that there is metric tons of contamination on the Turin Shroud with its veneration. I was told yesterday that people have kissed it, have handled it, have unrolled it. And DNA is ubiquitous. It's everywhere. I could take a vacuum one of these squares, and then look for genetic signatures and come up probably with hundreds of individuals. In building some clean labs for ancient DNA analysis, we came across a literature that said that you shed an unbelievably large number of particles every hour. And if you clean your glasses in the morning and then look at those an hour later, you can see that there are epithelial cells. So we're constantly shedding DNA. So the real challenge here is how do we, clean samples? And I was happy to hear from the last presentation, from Bob's presentation, that it sounds like sticky tape samples can actually be cleansed. So is there a mechanism there for cleaning sticky tape samples that have blood? So the challenges of ancient DNA analyses, this is supposedly some pottery that dates from the time of Nehemiah, is that like any venerable artifact, it's in pieces, because that's what happens over time. So there are challenges to ancient DNA analyses, but that doesn't mean that they cannot be overcome. And I would just like to make a public service announcement that DNA work is not like you see on crime scene investigations. It just doesn't happen in an hour. Our Titanic project actually took 7 years with the help of AFDIL, the Armed Forces Identification Lab, and several other labs for validation as well. So when you get involved in one of these, it's a bit of a marathon. So the DNA is degraded, but there are actually now tools and techniques that forensic labs use to actually amplify or replicate small pieces of DNA. So what we can do with DNA now, if you kind of think of it like a copy machine. So I can put a piece of DNA into this machine and I can copy it. And then I can read the sequence on there of the 4 basic letters of the genetic alphabet and look for these names and these themes that tell me the ancestry of this particular person or the population that he came from. Each one of you is incredibly a veritable source of genetic genealogical information, and that's the case with people in antiquity as well. So reconstructing ancient genetic signatures, we would do what's called profile Y chromosome with mini DNA fingerprinting techniques. So this is what the FBI uses now, DNA fingerprinting. They've reduced the size so that you can copy this degraded DNA. You can profile mitochondrial DNA with molecular techniques designed for ancient DNA studies. So this has been proven. This can be done. However, you have to use both comparative ancient and modern data sets to place results in both spatial and temporal context, which means that the data has to make sense. So if I have someone that is probably Hispanic in origin, you know what, that just simply does not make genealogical sense for what I'm looking for. So there are comparative data sets. And the work must be done in an ancient DNA facility or laboratory to ensure the quality control and quality assurance so that the results that are maintained are believable. So that's really what you're fighting against whenever you do any one of these projects is there contamination? Does it make sense? Have you taken the precautions? So for any samples, there must have an excellent, what they call chain of custody. It has to be excellent provenience. You have to know where that sample was, who had it, so on and so forth. And in this case, it must be large enough to ensure an appropriate, defendable endpoint. And this goes back to the sample size once again, that the DNA yield must be within believable parameters. So if we look at a sample, there must be enough cells there for me to say, yes, I can show you with my analyses and the spectra that there is actually DNA there. I know some of it is contamination because this artifact has been around for 2,000 years, but I know that I have nucleic acids. Again, the ancient signature must be present and convincing in spite of contaminants. So you're going to get a zoo of materials, but if you can show that the major portion here is actually this individual with these genetics that matches the time period and the suspected population, then you have to consider this as being authentic if everyone else looks sort of European. And again, the results must be consistent with population expectations and data from the region and the period. So I probably didn't show you too many neat pictures of the polymerase chain reaction or how we sequence the genome or things like that, but these are a list of recommendations that I think would lead to a result that would be believable and defendable, because I think, once again, I'd like to emphasize that bad results are really the worst kind of results. In ancient DNA, that can happen. A very good colleague said that he got DNA from a dinosaur, and it was published in a high-profile journal, but that was wrong, and that has sort of followed his career. We had some initial difficulty in identifying the unknown child on the Titanic disaster. And so you have to be very, very careful in how you do this. Again, it's just not a CSI project. So in summary, the sticky tape that I was sent is consistent with blood. There are molecular tools available for genetic analyses, and an adequate sample with some of the techniques I think that we've heard in a previous talk hopefully should enable DNA testing. So thank you very much.